Okay. Good morning, students.
Good morning, sir.
Good morning, sir. Good morning, sir.
Good morning. Only 83 are there. Before starting the class, please clarify your doubts regarding the test. Don't bother about the what is it average and other things. For internal assessment calculation is different. Final internal assessment calculation means we are supposed to give mark for 2 IA1, IA2 and one model. That is the split up. That too, we have different weightage for each. First IA weightage will be less. Then where moderate for second, model you will be having more percentage. Okay. Now and then, if anybody asks, dean or administration ask, means we will give the average of all the tests what we are conducting now. Okay. We can give all the 6 marks to the administration. So the purpose of putting average for general calculation is different. IA calculation for your final exam is different. Don't worry about all those things. That is our headache. Okay, if the test is announced, please write the test. Put your effort. Try to put your maximum effort. Try to get more marks. Are you clear? Any other doubt regarding the test? Yes, only is around 100 students are there. We'll wait for another 2 or 3 minutes. Anything else you want to clarify? Same day of theory, afternoon you will be having Viva. Here, Viva means you will be provided with the specimens. One from thorax, one from head and neck, next the bones. So questions will be asked. So it will be like a discussion. So that is there on the day of theory examination. Next, followed by that Thursday, you will be having spot test. That is spot test. So physically you are going to attend. So similar to our online spot test, you are going to see that specimens here. You are going to identify it. Anything else? Don't ask about average calculation and all. Other than that, no doubt, isn't it? So your duty to read. I am not
Excuse
happy
me,
about
sir.
that. Yes.
Sir, before our spotter test, will we have a revision for spotters all together?
I will make arrangement. Okay. Your thorax and head and neck. One day one day during your practical class, you can have the revision. That we will make arrangement.
Thank you, sir.
Thank you. Theory, first time you are going to give physical test, it seems. Have you written already physical test?
No, sir.
No, sir.
No. Okay. First time you are going to give. We have very big lecture hall, 300 capacity. So we can maintain physical distance and write the exam. So it will be arranged in new lecture hall. So participant 153.
Sir, your mic
Sir,
is
you're
off.
muted.
So today we are going to see about the development of central nervous system as introduction to our central nervous system. So already in the general embryology class, we have seen about the development of neural tube. Today, further what happens to that neural tube, we are going to see. So that is important for your introduction to central nervous system anatomy class. So competency 64.2, describe the development of neural tube, spinal cord, medulla, pons, midbrain, cerebral hemisphere, and cerebellum. Another competency 64.3, describe the various types of neural tube defect. Neural tube defect. So that only we are going to see here briefly. Then I will tell about the introduction to central nervous system. So that already you may be familiar with that. So first, formation of the neural tube. So see this picture. So already you are familiar with these structures. So our ectoderm, then our endoderm. So in between that two, we have that intraembryonic mesoderm. Basically, we have seen all these things. Then we have studied about the formation of notochord. So what is the function of notochord we studied? Function of notochord. Anybody, can you recollect? Functions of notochord. So you forgot. So these are all one or two things you can remember. So we are in detail we are reading about the formation of notochord. Then we used to tell about the function of notochord. It is the considered as primary organizer for the development of nervous system. Primary organizer. That means it stimulates the ectoderm present dorsal to that notochord. So this part of ectoderm present dorsal to the notochord is called as neuroectoderm. So what you have to remember till your life, your development of central nervous system is from ectoderm. The part of ectoderm that gives rise to brain and spinal cord is called as neuroectoderm. So that neuroectoderm is getting stimulated by the notochord. That's why notochord is considered as the primary organizer for the development of brain. If there is any defect in the formation of notochord, you will be having defect in the development of nervous system, defect in the formation of neural tube. So that only we are going to see now. So if there is any defect in the formation of neural tube, that only called as neural tube defects. Neural tube defects. Okay. So now this is the part of ectoderm called as neuroectoderm. So this is notochord. So now you see what happens to the neuroectoderm in the beginning of formation of central nervous system. So this notochord stimulates to proliferation of that neuroectoderm. So that proliferation of neuroectoderm leads to thickening of the neuroectoderm. So the thickening of neuroectoderm is called as neural plate. So now neural plate is formed. How it is formed? By the proliferation of neuroectodermal cells. So next what happens you see. So at the center of the neural plate, we have formation of a depression. So after the formation of depression, that neural plate is called as neural groove. Neural groove. Now you see these two are the margins of the neural groove. So at the margins of neural groove, some of the neural crest cells, some of the neuroectodermal cells are getting separated to form another group of cells called as neural crest cells. So these are all neural crest cells present on either margin of the neural groove. Later stage, what happens, how the neural tube is formed from the neural groove means you are having fusion of the margins of neural groove. We are having the fusion of margins of this neural groove that leads to formation of neural tube. Once the neural tube is formed, it will be getting separated from the neuroectoderm. At the same time, we are having that separation of neural crest cells also. So on either side of the neural tube, we have neural crest cells. Neural tube has been formed under the influence of notochord. So please don't forget, the function of notochord is the stimulation of neuroectoderm to form neural tube, but further differentiation of neural tube into brain and spinal cord. So that is the function of notochord. After that, it completely degenerates, except what are the remnant of notochord? Very good, very good. Nucleus pulposus. Anything else?
So the annulus fibrosis.
Annulus fibrosis. No. Only nucleus pulposus, not annulus fibrosis. Okay. Next, anything else? As you are reading head and neck now.
Ligament of dense.
Ligament of dense or dense itself. What is dense?
It will be in the axis, sir.
Very good, very good. So axis is the second cervical vertebra. Actually, if you take your first cervical vertebra called as atlas, it's not having body. But the axis vertebra, second cervical vertebra, is having a longitudinal projection called as dense or odontoid process. Dense or odontoid process. So that is considered as another remnant of our notochord. Next, you will be having attachment of a ligament to that dense. That is also considered as your remnant of notochord. As UG level, if you say nucleus pulposus, that much is enough. Okay. So now this neural tube is formed. That neural tube extends from cranial end to the caudal end of the embryo. So you'll be having formation, presence of this neural tube on the dorsal aspect of embryo from the cranial end to the caudal end. So imagine like that. So now the part of neural tube present at the cranial end further differentiate to form the brain. What you have at the caudal end forms our spinal cord. Now this is the dorsal view of embryo to understand the formation of neural plate and neural groove. So we have cut the wall of amniotic cavity. So cut edge of amnion you are able to make out. We are seeing the embryo from above. So regularly we are seeing that picture. So this is the dorsal view of embryo. Now this is the neural plate that is formed by the proliferation of neuroectodermal cells. Now center depression takes place in that. That is the neural groove. Later on, what happens? The margins of the neural groove fuse to form the neural tube. So now you see the next picture. So the beginning of formation of neural tube. So this is the complete formation of the neural tube. So important point, what you have to note here, note that the fusion of the margins of the neural groove takes place in the cervical region. From the cervical region, it ascends upwards and downwards. So the fusion of the margins of the neural groove is not taking place at the cranial end or caudal end, then it is further progressing. So that fusion of that margins of the neural groove takes place in the cervical region. From the cervical region, that is from the level of neck, it goes up to the brain level. Next, it comes down to the level of spinal cord. So that's what that fusion of margins of the neural groove takes place to form the neural tube. Now clearly you are able to make out that. So this is thickening neural plate. This is the central depression neural groove. Now the neural tube is getting formed. Now you see complete neural tube you are able to make out, but you are having a opening at the cranial end and another opening at caudal end. The opening what you have at the cranial end is called as cranial neural pore. What you have at the caudal end is called as caudal neural pore, or otherwise this is anterior neural pore, this is posterior neural pore. So that is important for MCQ. So already I told neural tube defect. So if there is any defect in the fusion of the margins of the neural groove, that leads to neural tube defect. At the same time, after some time, this anterior and posterior neural pores will be completely closed. Okay, then only we'll be having further differentiation of the neural tube to form our brain and spinal cord. So failure of closure of this neural pore also leads to neural tube defect. Sometimes this may be normal. By normal fusion of the neural grooves, the neural tube may be formed, but there may be failure of closure of the anterior or posterior neural pore. That is also coming under the heading of your neural tube defect. Okay, so now the neural tube has been formed. So we have two neural pores, anterior neural pore or anterior cranial, both are same. Caudal and posterior, both are same. So now you are going to see the brain specimens. Note that point now. Later on, try to clarify it with the teachers. Lamina terminalis. So when you are reading about the sagittal section of brain, ask the teachers to show lamina terminalis in the brain. That is the level of anterior neural pore or anterior end of the neural tube in adult brain. Lamina terminalis. So remaining part of the brain are getting differentiated from that level. Okay, so that is lamina terminalis. What we are seeing in the adult brain is the represents the anterior end of the neural pore, your anterior, otherwise anterior neural pore. If there is failure of closure of anterior neural pore, that leads to a condition that is called as anencephaly. So that we will see at the end of this class. I will explain about it. Now you see what happens to that neural tube. So neural tube present at the cranial end or cranial end of the embryo undergoes dilatation to form brain vesicles. So cranial part of the neural tube forms your brain. Caudal part of the neural tube forms your spinal cord. Simple diagram to understand that. So now what happens to that cranial end of the neural tube? So that cranial end of the neural tube is divided into brain vesicles, or actually it is getting converted into brain vesicles with three different parts. The cranial part is called as prosencephalon, middle part is called as mesencephalon, your caudal part is called as rhombencephalon. So you are familiar with this, isn't it? Prosencephalon, mesencephalon and rhombencephalon?
Yes, sir.
Yes, the prosencephalon represents forebrain. Mesencephalon represents midbrain. Rhombencephalon represents hindbrain. So that is the introduction to your brain. So if you take your brain, we have forebrain, midbrain and hindbrain. Forebrain is nothing but prosencephalon. Mesencephalon is nothing but our midbrain. Your rhombencephalon is nothing but hindbrain. So as an introduction, you should know this, important for your MCQ. Prosencephalon, mesencephalon and rhombencephalon. So these are the three different parts of the brain vesicles before the final formation of our different parts of the brain. Prosencephalon, mesencephalon and rhombencephalon. Now you see what happens to that prosencephalon. So from the prosencephalon, we have lateral outgrowth. We have two lateral outgrowths from the prosencephalon. After that, for our lateral outgrowth, if you see the prosencephalon, it is divided into diencephalon and telencephalon. So after that lateral growth, central part of that forebrain is called as diencephalon. The lateral outgrowth is called as telencephalon. So these two are telencephalon. This is diencephalon. Now, if you come down mesencephalon, we have midbrain. There is no much change. Now, if you take your rhombencephalon, that is also getting divided into two: cranial metencephalon, caudal myelencephalon. Metencephalon and myelencephalon. This is important. Furthermore, important for your MCQ. Telencephalon. Okay, diencephalon, mesencephalon, metencephalon, and myelencephalon. So what is that telencephalon? The two lateral outgrowth from the prosencephalon, that is nothing but our telencephalon, that only forms our cerebral hemisphere. So how many cerebral hemispheres we have?
Two cerebral hemispheres.
Yes, very good. So the telencephalon forms two cerebral hemispheres, important for MCQ. What about diencephalon? Diencephalon is in the median plane that forms mainly thalamus and hypothalamus. Thalamus, hypothalamus, metathalamus, epithalamus. So like that we have various parts of diencephalon. Important one is thalamus and hypothalamus. Already, what about physiology? Is it going on? Thalamus classes are? Thalamus, hypothalamus? Not yet. Over.
No, sir.
No. Oh,
Just the
okay.
introduction part.
Very good, very good. Thalamus, hypothalamus, metathalamus, epithalamus. Just be familiar with the terminology. Later on, you will read about it. But in physiology, it is very important. You are going to read a lot about central nervous system in physiology. So diencephalon, mainly thalamus, hypothalamus. Next, mesencephalon forms your midbrain only. Next, your rhombencephalon is divided into two: metencephalon and myelencephalon. Already I told rhombencephalon forms which part of the brain?
Hindbrain.
Hindbrain. Any idea what are the components of hindbrain?
Pons.
Yes, very good, very good. Pons, medulla, and cerebellum. Pons, medulla, and cerebellum. But now your rhombencephalon is divided into metencephalon and myelencephalon. Now, what are all the parts of brain derived from metencephalon? Our pons and cerebellum. Your myelencephalon forms your medulla. All these things are important for MCQ. Myelencephalon forms medulla. Metencephalon forms your pons and cerebellum. So that is the differentiation of your prosencephalon, mesencephalon, and rhombencephalon. Prosencephalon forms diencephalon and telencephalon. Mesencephalon, there is no further much differentiation, so it represents our midbrain. Next, we have rhombencephalon, that is divided into metencephalon and myelencephalon. Metencephalon forms your pons and cerebellum. Myelencephalon forms your medulla. So that's what we are getting various structures from the brain vesicles: prosencephalon, mesencephalon, and rhombencephalon. So this original arrangement will be like this only: cranial to caudal. We will be having prosencephalon, mesencephalon, and rhombencephalon. So this is the order. But finally, how we are getting our final position of your cerebral hemisphere, pons, medulla, and cerebellum, that we have to see. So how we are attaining that? So due to formation of fluxes, so that fluxes are nothing but your bend. Some bends are formed in that arrangement of brain vesicles to form our final form of the brain. So see that. So your prosencephalon, mesencephalon, and your rhombencephalon. So rhombencephalon divided into metencephalon and myelencephalon. So this is medulla. Medulla continuous as spinal cord. Am I correct? Medulla continuous as spinal cord.
Yes, sir.
Yes, sir.
Yes, very good. So another important point, have you seen foramen cecum? Sorry, foramen magnum?
Yes, sir.
Yes,
sir.
Foramen magnum. What is passing through the foramen magnum? Medulla. So that point you should be very clear. After passing through the foramen magnum, medulla continuous as spinal cord. So what is passing through foramen magnum is not spinal cord, your medulla. So your medulla, after passing through foramen magnum, continuous as spinal cord. So at that site, At the site of junction of medulla and spinal cord, we have formation of first flexure that is called as cervical flexure, important for MCQ. So that is the flexure to be formed at first is your cervical flexure. So one bend occurs at the junction of medulla and spinal cord. That is called as cervical flexure. Flexure already you are familiar, isn't it? Already we studied about hepatic flexure, splenic flexure, right colic flexure and left colic flexure. Is like that? It is flexure means bend. So next flexure what is formed is at the level of your mesencephalon. So that is called as mesencephalic flexure. Mesencephalic flexure, that is at the level of midbrain. We have the next flexure that is called as mesencephalic flexure. So next one what is formed is your pontine flexure. That is at the junction of metencephalon and myelencephalon. Another flexure is formed that is called as pontine flexure. So first flexure is cervical, next one is mesencephalic flexure, third one is pontine flexure. So after the formation of all these flexures, finally we are getting our final form of brain. Now you imagine, already I told what are the structures we are getting from prosencephalon, mesencephalon and rhombencephalon. So after the formation of these flexes, we have final form of brain. So if you know this much, that is enough for you. So what are the different parts of your brain vesicles? How we are getting that final form due to the formation of the cervical, mesencephalic and pontine flexure. Final flexure to be formed is called as telencephalic flexure. Final one is telen, first one is cervical. So after that we are getting this final form of brain. So see that, so this is your cerebral hemisphere at the center, so that is represents the telencephalon. At the median plane, we have diencephalon representing your thalamus and hypothalamus. Next, if you come down, we have our mesencephalon, that is nothing but our midbrain. Then if you come down, we have metencephalon, that is your pons and cerebellum. Next, we have our myelencephalon, that is our medulla. Now, what is brainstem? What are the components of brainstem, students? Brainstem.
Pons were
Pons
alive.
and medulla.
Hmm.
Pons, medulla and midbrain.
Right, very good. Above downwards, mesencephalon, your pons, not entire metencephalon, pons of metencephalon and your medulla. So this one, your midbrain, nothing but mesencephalon, then pons and medulla. So this is called as brain stem. So that is also important for your MCQ. So that's what we are getting our final form of the brain. So telencephalon forming your two cerebral hemispheres. Next, diencephalon forming your thalamus, hypothalamus, metathalamus, and epithalamus. Next, mesencephalon forming your midbrain. Next, metencephalon forming your pons and cerebellum. Next, your myelencephalon forming your medulla. So after these flexes, we are getting the final arrangement like this. Now, one more terminology what you should know is brain stem. Brain stem, that is above downwards, midbrain, pons and cerebellum. So this brain stem is connected to cerebellum, through cerebellar peduncles. Have you heard about it? Cerebellar peduncles? No. So superior cerebellar peduncle connects your midbrain and pons. Middle cerebellar peduncle connects your pons and cerebellum. Inferior cerebellar peduncle connects your medulla and cerebellum. So midbrain is connected to cerebellum through cerebellar peduncles. Note that cerebellar peduncles. Now important point. Already you are familiar with that. What are the various parts of the brain vesicle? What are all the structures derived from each? Now for each part of that brain vesicles, you have cavity. So those cavities are called as your ventricles of the brain. Ventricles of the brain. Now you see, so already I told from the forebrain, the center part diencephalon. On either side, what we have is the telencephalon. So now you see this telencephalon, already you know that it forms your cerebral hemisphere. What is that cavity of telencephalon? Is lateral ventricle. So we have two lateral ventricles, one on each side or one in each cerebral hemisphere. So that is lateral ventricle. Once again, important for MCQ. Cavities of each part of the brain vesicle is important for your MCQ. So cavity of telencephalon is lateral ventricles. So next cavity of diencephalon. So that is thalamus and hypothalamus is the third ventricle. So if you know this, you can write the boundaries of your ventricle. So that is also important. So now, what are all the boundaries of you expect for your third ventricle? From this picture, can you analyze and tell? Thalamus and hypothalamus. So third ventricle is only one third ventricle we have, but we have two lateral ventricles in each cerebral hemisphere. We have one, totally two. But if you take your third ventricle, only one third ventricle we have. So already you know that diencephalon forms your thalamus and hypothalamus. So lateral boundary of third ventricle is formed by your lower part of thalamus, and below that we have hypothalamus. So in between thalamus and hypothalamus, we have our third ventricle. So next you see that what are the other ventricles you know? Lateral ventricle, third ventricle,
Fourth ventricle.
Fourth ventricle. So now this is the fourth ventricle. Fourth ventricle is the cavity of rhombencephalon. So rhombencephalon already you know, it is divided into metencephalon and myelencephalon. Metencephalon forms your pons and cerebellum. Myelencephalon forms your medulla. So in between your pons, cerebellum and your medulla, we have our fourth ventricles. Now we see, so this is your pons, this is your medulla, this is your this is your cerebellum, this is your medulla. So now this is the site of fourth ventricle. So boundaries of fourth ventricles are found by pons, medulla and cerebellum. So that is introduction. So you are going to read about your fourth ventricle in detail. So where you should know about which are all the parts of brain form bounding that your ventricles. Then only you can write about the boundaries of the ventricles clearly. So once the fourth ventricle is over, clearly you have to write what are all the structures of pons forming your actually that pons and medulla forms floor of the fourth ventricle. Cerebellum forms roof of the fourth ventricle. Don't confuse those things now. Yeah, at the end of this class, if you know that your pons, medulla and cerebellum forms boundaries of your fourth ventricle, that much is enough. At the end of this today's class, at the end of the brain class, you should be able to clearly write about your cerebellum forms your roof. Your floor is formed by partly pons and partly medulla. So that's why pontine part of flora, flora of fourth ventricle, medullary part of flora of fourth ventricle. That's what you are going to read. So you are separately, you are going to read about pons, medulla. So we are going to start from spinal cord. So you'll be having class of spinal cord, pons, medulla, cerebellum. So after completion of all these four classes, you will be reading about fourth ventricle. So at that time, you should be able to clearly write about that flora of fourth ventricle. That is very important essay question for you. So now you see this picture. What is the communication between your third ventricle and fourth ventricle? Yes, students, see and tell.
Cerebral aqueduct.
Cerebral aqueduct. So that cerebral aqueduct is the cavity of mesencephalon. So all are important for your MCQ. So cavity of telencephalon is the lateral ventricle. Cavity of diencephalon is the third ventricle. Cavity of rhombencephalon is the fourth ventricle. Now the cavity of midbrain or mesencephalon is cerebral aqueduct. Cerebral aqueduct. Already you know that what is the use of having ventricles. So your cerebrospinal fluid is formed in this ventricle. From this ventricle, it is finally reaching the your subarachnoid space. Subarachnoid space. So there only our main major amount of your CSF will be there in the subarachnoid space. So from the fourth ventricle, it reaches the subarachnoid space. So first the CSF, what is formed in the lateral ventricle, will reach the third ventricle through this opening. So this opening is between your lateral ventricle and third ventricle on either side. So as we have two lateral ventricles, we have two openings here. Through that openings, your lateral ventricles are communicating with the medianly placed your third ventricle. So that opening or foramen is called as interventricular foramen. Any other name is given? Interventricular foramen. Foramen of
Foramen of Monroe.
Monroe. Very good. Foramen of Monroe is the name given to that interventricular foramen. As it is between lateral ventricle and third ventricle, it is called as interventricular foramen. Now, third ventricle is connected to the fourth ventricle through cerebral aqueduct. So this is the cerebral aqueduct. Through that, your CSF from the third ventricle comes to the fourth ventricle. Now, if you trace this fourth ventricle, it is in continuation with the central canal. So central canal we have in the lower part of medulla, then it continues in the spinal cord. So this central canal is there in the spinal cord also, but very minimal CSF will present in that central canal. But from the roof of the fourth ventricle, so later on you are going to read about it. So you will be having openings. Through that, your CSF from the fourth ventricle will reach the subarachnoid space. Subarachnoid space. So from the ventricle, it reaches the subarachnoid space. There only the main circulation of your CSF takes place. If you trace your fourth ventricle down, it is continuing a central canal where we have very minimal amount of CSF only. Okay, so that is the your cavities or ventricles of the your brain vesicles. Very important for your MCQ. For introduction also it is important. Where we will be having, now you are having a mental picture. Two cerebral hemispheres are there on either side. If you go to the median plane, we have thalamus and hypothalamus. Continuous down, mesencephalon, that is your midbrain. Then your metencephalon, pons and cerebellum. Next your myelencephalon medulla. So that is the arrangement. That mental picture you should have now. Now the ventricles, two lateral ventricles are there in the cerebral hemisphere. Both are connected to that centrally placed single third ventricle. From the third ventricle, it is going to, your CSF is going to fourth ventricle. So through aqueduct of Sylvius. So now this is that your basic things you should know about the development of brain with your introduction to CNS. So next important thing is derivatives of neural crest cells. So already I told what is that neural crest cells. Neuroectodermal cells present on either side of the neural groove. After the formation of neural tube, they are getting separated and present on either side of the neural tube. Main structures derived from the neural crest cells, important for your short notes, derivatives of neural crest cells. So postganglionic sympathetic neurons, Schwann cells. So already you know, so as you are reading the CNS in physiology. Next, your dorsal root ganglion. So dorsal root ganglion. So what is the dorsal root ganglion? What do you know about it? Dorsal root ganglion. Spinal cord is over, you know.
Spinal cord posted dorsal side of it.
That's okay. That's why it is called as dorsal root ganglion.
Sensory
That means it
nerve.
should be there in the dorsal root. What about spinal cord is over in physiology?
So it's still
Spinal
going
cord.
on.
No, sir.
Still it is going on. So it is a very long class. Okay, so you are reading about ascending tracts, isn't it?
Yes, sir. Ascending and descending.
Ascending and descending tracts.
Yeah, very good, very good. So as you know about ascending tract, what you know about dorsal root ganglia?
Okay, give the sensory nerves.
Still
The
it
first
is
order sensory
only.
neurons.
So the first order
neurons.
neurons terminate.
Very good, very good, very good. That is important. That is the answer I expect. Okay, so first order neuron, this and all simple thing. It is present in the dorsal root. So dorsal root ganglion. Dorsal root is sensory in function and all very basic. Motor root, your ventral root is motor in function. Dorsal root is sensory in function. Even though it is mixed, predominantly dorsal root is sensory. Predominantly motor root is your ventral root is motor in function. This and all very basic. So if you tell first order neuron for the ascending tracts are there in the dorsal root ganglion. Very good, very good. Some of the students have understood the tract. Okay, so like that for all the ascending tracts, please read about first order neuron, second order neuron, third order neuron. Any tract is having first order neuron in the brain. So I will be very happy if you answer for that. All the ascending tracts are having first order neuron in the dorsal root ganglion. Any other neuron having it in the brain or in the medulla? Yes, Pranav?
Dorsal column.
Sir, no ideas.
No idea.
remember.
Okay, somebody is telling dorsal column tract. That's what you want to tell? Is somebody committed? Please commit. What's your name?
Annie, sir.
Yes, very good, very good. So we have tracts in the posterior funiculus or posterior column tract. What the name?
Nuclei, sir.
No. What I asked is which tract is having first order neuron in the medulla, or otherwise we used to ask as brain.
Tract of
Dorsal.
Hall and Burdak or tract of
Gracilis,
unique.
Gracilis and
Gracilis.
cuneatus. Gracilis and cuneatus. These are the tracts present in the dorsal funiculus, or they used to call it as dorsal column tract. Isn't it? They are carrying proprioceptive impulses from the body. So here the relay of these neurons takes place in the medulla. Gracile tubercle and cuneate tubercle are there in the medulla. So first order neuron for relays in that gracile tubercle and cuneate tubercle present in the medulla. For all other tracts, ascending tracts, the first order neuron is there in the dorsal root ganglion. So that is the difference. Very good, very good. So please you understand and read about all the tracts. It will be easy for you. So you can remember. So during your final year medicine exam, definitely they will ask about these ascending and descending tracts. So please understand and read, then it will be easy for you. So that is the your dorsal root neurons are derived from neural crest cells. Next, your pia and arachnoid matter yet to be proved, but as of now you can say like that. But pia and arachnoid matter are derived from your neural crest cells. Next, your adrenal medulla. So adrenal medulla, not cortex. Okay. Next, dendritic cells of skin. Any name is given to those cells? Dendritic cells. Dendrite. Which cell of your skin is having projections like dendrites? Melanocytes. Langerhans cells. We have Langerhans cells and melanocytes. They are having projections like dendrites. They are coming under dendritic cells. Dendritic cells. Okay. So these are all these structures mainly derived from neural crest cells. In addition to that, one tabular column is there. Some of the other contribution also there from the neural crest cells. Connective tissue and bones of the face and skull. Contribution is there. They are not entirely derived from your neural crest cells, but some role is there from the neural crest also. Cranial nerve ganglia. So the sensory ganglia of the cranial nerves. Next, C cells of thyroid gland. So already we studied about spiral septum in the heart. Okay, so these neural crest cells are migrating and reaching the all these areas. There, its role is there. That's what it is proved out. Odontoblast, that is giving rise to your teeth. Dermis in the face and neck. Note that development of skin means you have epidermis and dermis. Epidermis is from ectoderm, dermis is from mesoderm. But your dermis of face and neck, it is contribution is there from the neural crest cells. Next, dorsal root ganglia, already we told. Sympathetic chain and pre-aortic ganglia. Parasympathetic ganglia of GAT, adrenal medulla, already you know. Schwann cells, you know. Glial cells, supporting cells of brain. Meninges, already you know. Melanocytes, already I told. Smooth muscle cells of blood cells of your face and forebrain only. This and all yet to be proved, but suppose if we ask short words, you can enumerate. 1 minute, please. So now, what is this spina bifida? That is nothing but neural tube defect. Neural tube defects generally called as spina bifida. See the picture A, B, C. What is the defect? Please find out.
Nose
So the
Vertebral arch is
subarachnoid
not
is upper and subarachnoid.
Yes. So here you see the spinal cord is covered only by skin. There is no vertebral arch. So here that neural tube defect is there. Complete formation of, so if you have complete formation of neural tube, the formation of vertebral arch will be there. It is not there. Simply everything is normal, and the dorsal aspect of your vertebral column is not there. But of course, we have outer covering of skin. But otherwise, everything is normal. Okay, so this is called as spina bifida occulta. So important thing, it is if in the sacral region and the median plane, if you have tuft of hairs like this, you have to suspect spina bifida occulta. Spina bifida occulta. So everything is normal. So spinal cord, your meninges are in normal. So only this defect is there. That's why it is called as spina bifida occulta. Nothing will happen. So everything will be normal. Okay. So that is the one minor form of spina bifida. So here what happens, that projection will be there at that particular site. It may be at any level, so not only at sacral level, but this is more common in the sacral level. The reason we don't know. So in the sacral level, if you have tuft of hairs in the site of your vertebral column, you have to suspect this. So next, here we have, what is abnormal here?
Subarachnoid space.
You have herniation of meninges. So this is called as meningocele. That's why it is called as meningocele. So your meninges along with subarachnoid space is herniating through the defect. So that's why it is called as meningocele. So in this case, that definitely that projection will be there at that particular site where we have that defect. Now this is called as meningomyelocele. Meningomyelocele. So here we have, along with your meninges,
Spinal
we have herniation of spinal cord also. That's why this is called as meningomyelocele. So like this, you imagine if there is any defect at the occipital region, what will happen? Instead of spinal cord, you will be having part of brain. Am I correct?
Yes.
Yes, sir.
Yes, sir.
Yes, that's what you have to understand. It may be at any level. So what I am projecting now is the at the spinal cord level. So this is very, very rare, very rare condition that is called as rachis. Rachis. So what is the defect here? Find out.
Neural tissue
the
is
neural-
Tissue is
That's
not
not fused.
Only neural,
Neural plate is not formed.
only
Sir,
Spinal cord
neural
is not there.
plate. You have formation of neural plate. After that, neural groove has been formed. After that, nothing has been formed. Okay, neural groove is there. That neural groove is getting folded. So there is no neural tube formation at all. So this condition is called as rachis. So these are all the various grades of your spina bifida, commonly called as our neural tube defect.
So the child
this-
won't survive, right? For the rachis.
Rachis is no chance, no chance. So this and all you can have. So this may be meningocele or meningomyelocele. This and all we can do correction. Meningocele or meningomyelocele. So this is at the level of thoracic region, spina bifida, thoracic region. So same thing in the brain, level of brain means meningocele, meningoencephalocele. In case of spinal cord, we are telling meningomyelocele. Here encephalocele. If the ventricle also herniating, that is called as meningohydroencephalocele. That word encephalo denotes brain. That word myelo denotes spinal cord. So that's what we have seen here. Meningomyelocele. Instead of myelo, if you put encephalocele, that denotes brain. That word hydro comes means including the ventricular part, it is projecting through the defect. Meningoencephalocele. So this is the condition called as anencephaly. See that your scalp is not there, cranial vault is not there. So your brain got atrophied due to the exposure of brain to the amniotic fluid. So this condition is called as anencephaly. That is due to failure of closure of anterior neural pore. Anterior neural pore. Already I told, not only the failure of fusion of margins of the neural groove leads to your neural tube. So failure of closure of anterior neural pore also coming under the neural tube defect, anencephaly. Okay, so this is called as anencephaly. Okay, so important for your MCQ. So in our museum, we have this specimen. Okay, anencephaly specimen is there, fetus is there. Please go and see. So this is anencephaly with spina bifida. Anencephaly with your cervical and thoracic region. We have spina bifida. Okay. So these are all the few things you should know about the your spina bifida or neural tube defect. So one more PPT is there. There's few words about the your cerebral hemisphere and other things.
So,
Yes.
sir, can all these defects be corrected, sir? Like the child will
Depends
become-
upon the degree. Meningocele, meningomyelocele and all easily we can do. When we are diagnosing antenatally, it's not possible. Postnatally, it's possible, but it depends upon the degree. So now you see, so this is the superior view of brain, what you are going to see now. So superior view of two cerebral hemisphere. What is this called as? That is separating the two cerebral. Pardon?
Corpus callosum.
So the sulcus and
This is a guru. Separating the two. Corpus callosum is connecting the two cerebral
The
hemisphere.
cerebral cortex.
Median longitudinal fissure. Median longitudinal fissure. What is lodged in it? Related
The
theory is
false
over. That's why I'm
cerebri.
asking.
False cerebral.
Pardon? Fox cerebri. Very good. Very good. This is called as median longitudinal fissure that separates two cerebral hemisphere. Once you get your brain, try to see that. If you separate the two cerebral hemisphere through that median longitudinal fissure, at the floor of the median longitudinal fissure, you can see one arched structure, that is corpus callosum. Now you see, so this is your falx cerebri. So this falx cerebri is lodged in this median longitudinal fissure separating the two cerebral hemispheres. Now this is one cerebral hemisphere. So this is cerebellum, this is pons, this is medulla. So then each cerebral hemisphere, if you take, they are divided into 4 lobes. Your frontal, parietal, occipital, and temporal. So frontal lobe, your parietal lobe, occipital lobe, and temporal lobe. Already you may be familiar with all these things. How we are dividing it into various lobes and all, you are going to learn. Now this is the sagittal section. So already I told, this is the plane of median longitudinal fissure. Median longitudinal fissure. So if you separate the median longitudinal fissure gently, like a very 1-day-old baby, you have to handle the brain. Okay, you should be very, very soft. So if you separate at the floor of that your median longitudinal fissure, so you will be having corpus callosum. So this is the corpus callosum. So this is the superior surface of corpus callosum that you can make out. So this is corpus callosum. Now in this median sagittal section, so you have to see the ventricles. So already you know that this is your cerebral hemisphere developed from
Telencephalon.
very good. What is the cavity of telencephalon?
Lateral ventricle.
Lateral ventricle. See that? What is given in blue color? That is your lateral ventricle. So this is your lateral ventricle. Okay. So now you see, this is your thalamus. This is your hypothalamus. So these two are coming from where? Diencephalon. So what is the cavity of diencephalon? Yes, see that this is your thalamus, this is your hypothalamus. So in between thalamus and hypothalamus, what we have is the third ventricle. So this is your third ventricle. So now you see lateral ventricle is connected to third ventricle through
Cerebral aqueduct.
no, lateral to third.
Foramen of Monroe.
Foramen of Monroe. The arrow mark you are able to make out, isn't it? Yes. You are able to make out the arrow mark.
Yes, sir.
Are you able to make out?
Yes.
sir.
The arrow entering into the third ventricle through interventricular foramen. This and all important spot is for you. Okay, we will probe that interventricular foramen. We will ask its communication or boundaries. Okay, so now you see, so this is telencephalon cerebral hemisphere. This is diencephalon thalamus and hypothalamus. Now this is mesencephalon or your midbrain, only the small part. Now this is the aqueduct. So this is the
Sir.
cerebral aqueduct. Now this is your hindbrain. This is your hindbrain. So in continuation of your midbrain, what you have here is the pons. This is your medulla. Pons, medulla, cerebellum. Okay, so constitute your rhombencephalon. Pons and medulla called as metencephalon.
Metencephalon.
Metencephalon. Now this is myelencephalon. So this is the fourth ventricle. So what you see here between pons, medulla, and cerebellum is the fourth ventricle. Are you clear now? Lateral ventricle, third ventricle, aqueduct of Sylvius. This is your fourth ventricle. So aqueduct communicates third ventricle and fourth ventricle. Actually, this is called as roof of the fourth ventricle found by cerebellum. This is called as floor of the fourth ventricle found by pons as well as medulla. Dorsal part of pons, dorsal part of medulla. Now another terminology I told is brain stem. So your midbrain, pons and medulla. Midbrain, pons and medulla forms your brain stem. Okay, are you clear? So what you have seen here is the lateral surface of cerebral hemisphere. What you are seeing here is the medial surface of cerebral hemisphere. Not only cerebral hemisphere, entire brain. Okay, so this is the sagittal section. So these two are important for your spotters. We used to keep your lateral surface. So in this you are going to read about sulci gyri. So once you read sulci gyri, what are all the important functional areas present in sulci gyri you are going to read. So that's why simultaneously your classes are going on in physiology and anatomy. So please try to correlate both and read. Don't read separately the tracks in physiology and tracks in anatomy. Okay, are you clear, students?
Yes, sir.
So separately reading anatomy, brain, then physiology, CNS, that should not be like that. While reading about acentric tracts, you are reading means take the anatomy book also. See that way cross sections, various levels of your cross sections of brainstem and spinal cord and read. Then only it will be easy for you. Okay, so all the tracks you are reading here, of course, this is medulla, then spinal cord. From spinal cord, medulla, various levels, pons, actually for all the tracks you are going to read through the transverse section of medulla at 3 levels, 2 levels of pons, 2 levels of midbrain. So you are tracing all the tracks from the spinal cord, medulla, pons, midbrain, thalamus to cerebral cortex. So the arrangement you see thalamus, hypothalamus, midbrain, pons, medulla. So this you should keep it in mind. So while reading about the tracks, you should know about what are the parts of brainstem. So from the brainstem, it goes to thalamus, thalamus to cerebral hemisphere. So now this is the ventral view of brainstem. Ventral view of brainstem. So this is your ventral part of midbrain called as crest cerebri. Crest cerebri. Next pons. Next medulla. So this is medulla. This is pons. This is your midbrain. Crest cerebri. Crest cerebri means nothing but ventral part of midbrain is called as crest cerebri. Okay. So as of now, if you know, this is midbrain, this is pons, this is medulla. This is important for you. This is the ventral view of brainstem. Why it is important in anatomy means the attachment of cranial nerves. You are able to make out all the cranial nerves here. So this is optic, your oculomotor, trochlear, trigeminal, abducens, facial, your vestibulocochlear, your 9, 10, 11 cranial nerves here. Hypoglossal nerve is here. Hypoglossal will be here. Okay, so that's what all the cranial nerves are attached here in the brainstem. That is called the superficial attachment of cranial nerves. Superficial attachment of cranial nerves to the brainstem. Another thing, while reading about the tract, this arrangement is important. Already you told that first order neurons there in the dorsal root ganglion. From the dorsal root ganglion, it reaches medulla, pons, midbrain. Okay, next your thalamus, then your cerebral hemisphere. So this is the base of the brain or inferior surface of the brain. So already you have seen lateral surface, that we used to call it as supralateral surface. Next, your medial surface or sagittal plane or sagittal section of brain. This what you are seeing here is the inferior surface of brain. So your cerebral hemisphere on either side you are able to make out. This is the your median sagittal groove or fissure, median sagittal fissure. Next, this is your pons, this is your medulla, this is your cerebellum. So these three way only you are going to see the brain: supralateral surface, medial surface, and your inferior surface. So this is your cerebellum, superior surface of cerebellum. So this is called as cerebellar notch. There only you will be having lodging of your brain stem. Brainstem. So this is your midbrain. Then you will be having down pons and medulla. So these three are lodged in this notch. So this notch is called as anterior cerebellar notch. In the anterior cerebellar notch, you will be seeing this brainstem. Already I told brainstem is connected to cerebellum through cerebellar peduncles, superior cerebellar peduncle, middle cerebellar peduncle, and inferior cerebellar peduncle. So this is the arrangement of ventricles. So two lateral ventricles connected to third ventricle, aqueduct, fourth ventricle. So this is coronal view. So this is your lateral view. So this is your third ventricle present in the cerebral hemisphere. Then your third ventricle present in the diencephalon. So here this is the third ventricle. So this is your interventricular foramen. Now this is your fourth ventricle. Here you will be having aqueduct. Now the blood supply of brain mainly from two sources. Already you studied about vertebral artery. So that is called as vertebral system. Next is your carotid system. Basically vertebral system and carotid system. Vertebral arteries, then carotid artery, that internal carotid artery continues as middle cerebral artery that gives rise to anterior cerebral artery. From the vertebral artery, we will be having posterior cerebral artery. So three cerebral arteries are there: posterior cerebral, middle cerebral, and anterior cerebral. Out of these three, your middle cerebral and anterior cerebral are branches of internal carotid, are part of carotid system. Your posterior cerebral is part of vertebral system. So this is called as vertebral system. So this carotid system and vertebral system are communicating with each other through a circle. So that circle is called as circle of Willis. So later on you are going to read in detail. Now if you know that we have, if you go to the arterial supply of brain, we have carotid system and vertebral system. From the vertebral system we have posterior cerebral. From the carotid system we have anterior cerebral and middle cerebral. So both carotid and vertebral system are connected together through a circle that is called as circle of Willis. Important short notes for you. Circle of Willis. So this is circle of Willis. So later on we can read. So like that we are going to read about your central nervous system. So first class, what we will be having after this introduction class is your spinal cord. See that? So real brain and spinal cord, what we have removed here. So this is your spinal cord. Okay, so these two cerebral hemispheres, median longitudinal fissure. So this is the median longitudinal fissure where we have lodging of your Fox cerebri. Fox cerebri. Okay. So with this idea, please read about your central nervous system. As I told already, don't separate anatomy and physiology for reading purpose. So you have to integrate and read. It will be easy for you. You will be getting time also. Don't say we don't have time to read anatomy of brain and the CNS of physiology. Okay, are you clear, students?
Yes, sir.
So you have got some idea to read your brain. So with these ideas, please proceed with that. Please try to read on the day itself. That's way beginning, from the beginning itself. I am telling, same day, if you are having 2 theories or 3 theories, means same day. Please just you go through whatever we are telling in the book. Superficially you can read. If you don't have time to read in depth, then only it will be easy for you. Whatever points we told, important points, you can note down and note down on the same day. Whatever important for MCQ we are telling, that also please note down. You can get more marks in the MCQ. So same day you have to read. So that's what you have to plan for your reading. How much time you are allotting for your reading, we don't know. So you should be active. There should not be any difference in your concentration between your morning classes and afternoon classes. Or no other go hereafter 8 to 4 throughout the course we will be having classes. Okay, mainly if you go to second year, third year, all major lecture will be in the afternoon only. At least now you have practical, but second year, third year and all afternoon you will be having main lecture followed by practical. Okay, morning and all you will be in the hospital. So no other go, once we selected medicine, no other go. What is needed is hard work. We have to allot more time for reading. Throughout life you have to read. This is must. Okay. So with this I will stop this introduction to CNS as well as the development of brain. So next class we will be having your pituitary gland. After that you will be having regular spinal cord. In the order only you will be having spinal cord, medulla, pons. While reading about spinal cord, pons and medulla, you have to read about the ascending and descending tracts. Then only it will be easy for you. Okay. Okay, students, we will leave.
Thank you, sir.
174.
Thank you, sir.
Thank you.
Thank you, sir.
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 Thank you, sir.